Hey everybody, so we are looking at section 14.2, which is about ribosomes and protein synthesis. So we are continuing along this pathway of discussing how we get from DNA sequences or genes to actual proteins and phenotypes, right? So remember in 14.1 we talked about what RNA is, how that differs from DNA, the three different types of RNA that you need to be familiar with this chapter. That would be mRNA, tRNA, and rRNA, and then also transcription. Now, transcription is going to precede where we're headed here in section 14.2. So with this protein synthesis process, you always have to have transcription occurring, and then you get to this next stage, which is called translation, and that's what we're going to discuss here. So with this section, we have three objectives. We're going to look at the genetic code and how we can go from a sequence of nucleotides and letters like A's, C's, G's, and U's into a language, a different language now of amino acids, which are the monomers, remember, that make up proteins. So we're going to look at exactly how that translational process happens. We're going to also talk about what the ribosome has to do with all this, and then kind of review those different types of RNA that I just mentioned a few seconds ago. So as a reminder, right, when we talk about all of this happening, this is all occurring as part of that central dogma. If you remember what that looked like from section 14.1, we were looking at starting out in the nucleus with DNA, taking that and creating a temporary copy in the form of mRNA through a process called transcription. And then from there on, we take that mRNA molecule once it's been edited in the nucleus, and we pull it out into the cytoplasm, send it to a ribosome, where we're going to go on and make our actual protein or our phenotype. So when we make these proteins, it requires the use of not only mRNA or messenger RNA, also rRNA or ribosomal RNA, and tRNA. So this process here in 14.2, this translation process, incorporates all the types of RNA that we discussed earlier. So when we talk about genetic code, we're looking at the sequence of letters represented in a gene. And remember, when we talk about DNA, that includes thymine or T and not uracil, U, which are being shown here. The image that you see on this screen is supposed to be a transcript or a temporary copy of a gene, right, that we would find in a messenger RNA molecule. We know that because we have this single-stranded molecule. It doesn't take on the shape of that tRNA that kind of looks like a T, and it certainly doesn't look three-dimensional like the ribosomal RNA that we saw in section 14.1. I also know that this is going to be an mRNA because it has these things labeled down here as codons. So when we talk about taking nucleic acid sequences like this one here and converting it into a polypeptide or a protein, we require kind of a key in a way to get from one language to the other. And that key is represented by these codons that you're seeing here. So the ribosome, as part of this process, and we'll see this in a minute, is going to read this mRNA sequence in groups of threes, okay? And so these groups of threes that are labeled here are what we call codons. They're essentially like codes that tell ribosome which monomers of a protein to add and where. So codon is a group of three nucleotide bases found only in messenger RNA that specifies a particular amino acid, all right? And so if we look at what that means, this one codon here is AUG. This one is called AAC, and this one is UCU. And you can see all that I'm doing is just reading the sequence of letters, right? But I could say in this sequence of 9 RNA nucleotides, I have a total of 3 codons, okay? So it's basically something that we're dividing into groups of threes. All right, and so these codons are going to be super important because these codons, again, like I said, are kind of that link that we're going to use, that the ribosome uses to connect a nucleic acid sequence to a protein or an amino acid sequence. So when we look at the possible combinations of A's, G's, C's, and U's in a molecule of RNA, there are 64 possible combinations. This right here that you're looking at that looks probably completely overwhelming is something called a codon chart. And this is scientists' way of kind of inferring and figuring out what each codon corresponds to in the form of a protein. So for example, if we have the codon CAC, and I kind of read it like this, I go from the interior to the exterior. So every codon that starts with the letter C, I would start here in this kind of quadrant here on the lower right. So if I have CAC, that codes for an amino acid down here called histidine. Remember from back in chapter 2 when we talked about biochemistry and introduced these types of molecule groups, we said that there are 20 different amino acids. Now I'm telling you here we have 64 possible codon sequences, okay? And so what that tells me is that there's going to be some redundancy here. And what I mean by that is if we look at histidine, there are two different options. Histidine is the same amino acid in a protein, but it can be coded for by CAC or by CAU, right? Proline, here's the next one. It can be coded for by CCG, CCA, CCC, CCU. So there are some that are kind of duplicated, but you'll notice that there are also some others where it's kind of one and done. You only get one option and that's it. And so tryptophan is one of those down here. UGG is the only codon that creates tryptophan. Okay, and same over here, methionine, AUG is the only potential codon that will add a methionine amino acid in a specific position in a protein. Okay, so this is kind of like, if you're in foreign language and you have like a translator of some sort, right? Or if you use Google Translate as an example, this is kind of doing that job, right, for a cell. All right. And in this case, this is how we interpret what a cell's doing kind of naturally and on its own. So here are some of those examples, right? I mean, we can see exactly how we kind of eliminate some of those options. So when we look at this whole chart, a couple of things should kind of stand out to you. I mentioned that there are some redundancies here, but in addition to that, we do have those few where it's just one single code that creates that one amino acid. Among these different codons, there's always one type of codon that serves as a start. So this means that every protein that's created in any living thing, whether it be prokaryote, eukaryote, plant, animal, bacteria, fungi, protists, whatever, they all start with the same exact codon. Okay, and so the codon that we use to start any protein is actually AUG. And AUG, as I just read to you, codes for an amino acid called methionine. You can see that down here. So methionine is going to start every single protein in every single living thing, and they remain there. It's something that all living things kind of share in common. And it's something that a ribosome, regardless of where it's found, will always look for as the starting point of this translation process. There are also 3 stop codons, and these do what it sounds like they would do. They're going to tell the ribosome when to stop the process of creating a protein. And those are UAA, UAG, and UGA. And so if we go over here and look, UGA is a stop, UAA is a stop, and UAG is a stop. Okay. Other than that, all these other strange names that you see here are all of the names of the 20 different amino acids. You certainly do not need to know all of these names or how to spell them or how to say them, sound them out. Really, you need to just be able to interpret a chart like this to identify for me what a specific mRNA sequence could code for. Okay, and so what I mean by that is something like this. So what I've done here is I've added this slide. This is not in the one that's up on Canvas yet. But this slide, what you're seeing is a DNA sequence. And I just created kind of a short and simple DNA sequence here. One of the things I want to bring up, this genetic code here is the one that you just looked at here on the previous slide. Okay, it's circular. Some students I know like to look at this, and this one makes more sense. But there's also a codon table that can look like this. In both cases, you always start by reading the first letter of the codon, so the one kind of on the left of that sequence, just like you're reading anything. And the first position here is everything here. So you can see if I find, let's see that I want to look for one of those stop codons. Let's find UGA. So my first position here should be a U. My second position here should be a G, and then A should be the next one, which it says stop. Now, this says T because that T is representing what we find in DNA, actually. But remember, our codons are actually found in mRNA. All right, so this is a little bit deceiving. On your test, you would only see a codon chart that included U's and not T's, all right, to make that a little bit simpler. But the first step here, if I were to give you a gene sequence and say, all right, tell me what the protein sequence is for this. What corresponds? What's my corresponding or complementary amino acid sequence? And so your first step is going to be to take that DNA and actually convert it into an mRNA sequence. So my mRNA sequence down here is what we've seen before. We're going to write it complementary to our original DNA. So if in DNA I have TAC, well, T will pair with A in mRNA. The A in DNA will pair now with U in mRNA, because remember, mRNA doesn't have a T, and C is still going to pair with G, and that continues on. So all of these are complementary, just like what we'd seen in the past with DNA and, the two sides of the double helix. It's just that now I have a U in place of where I would normally put a T. Okay, so this is kind of what that complementary mRNA would look like. And remember, this is what we would get out of transcription. Okay, the first step, the part that we talked about in 14.1. When I go on to make a protein, what I'm going to do is have to read these codon charts, right? So AUG in this case, I would go down here and I'd say, okay, AUG, which again, if I swap out that T for a U, I end up with methionine, okay? My next one, glycine, GGC. If I want to see that I did this, properly and check my work, GGC codes for GLY, which is the shorthand for glycine. Leucine is the next one, CUG. So what I did again, CU, which is again our T in this case, and G, that creates leucine. Isoleucine, AUC, which is over here, ILE is short for that. And then I ended with a nice stop codon. Now, truly, this is a whole lot shorter than proteins that we would have in a living thing. Usually those are hundreds or thousands of amino acids in length. But this protein here, we're going to just, for simplicity's sake, say that we're ending. And so I end up with UAG, which is right here, or a stop codon. So that's kind of how that translation process happens, all right? Now, and not necessarily how it happens, but how we would kind of figure out what the sequence represents, all right? We're going to talk about here briefly, what the steps are and what's actually happening in a cell to do this. But for our purposes, what this means is that if I give you a sequence, whether it be DNA or RNA, I should be able to ask you to then figure out the protein or amino acid sequence. And this is something that I know you're going to be doing through some gizmos and Savas and other things like that. So you will get plenty of practice with this. And we can, Zoom and review and do it in class if we need to as well. So you will get more practice with this. So if it feels like it's a little foreign right now, that's okay. Come back, re-listen, right? Write this down if that's helpful. And certainly don't let these two throw you off. You can go back and check my work if you want to go back to here and use this as your codon chart, okay? You can also Google codon chart and you can get any shape or type that you feel like would be best for you. I will add, you will have to do this on my test. I will give you a codon chart. You do not have to have one or jot it down on your notes. You will have it available to you on your laptops as part of the test. Okay, and the same applies for a quiz. If we have a question like this on a quiz, you're always going to be given a codon chart or table. And that also includes the state EOC at the end of the year. So if you're asked to do something like this, they will always provide that information for you. You certainly do not have to memorize any of this. And again, you don't even have to be able to say the name or even really write out the whole thing. In this case, these are just the 3-letter codes, right? And so that can be used. So in thinking about, the actual cell process behind all this, how we take this one language of nucleic acid and convert it into amino acid. Well, we saw, how we can do that on paper to kind of simulate what would be happening here. But we also want to talk about, what are the actual molecular processes? What are the steps that the ribosome takes to do all of this? And so where we're starting again here is just right after transcription, we're going to start this process of translation. We don't want to leave this temporary mRNA copy just hanging around for too long. They're not meant to kind of stay around forever, right? They're just temporary. So after we've transcribed and created our mRNA sequence, after we have edited that sequence and removed the introns that do not need to be there, that mRNA sequence can then kind of snake its way out of the nucleus through these little nuclear pores, and it's going to find its way to a ribosome. Okay, now ribosomes, remember, if they're freely floating, they're going to create proteins that will end up staying inside the cell. If it goes to a ribosome that's bound and stuck to the rough ER, it's going to create proteins that will eventually route through the Golgi and then through the vesicles to be pushed out of the cell. Okay, but in this case, our mRNA that we created here, our newly synthesized mRNA that came out of the nucleus, is going to be used as our template to create a protein, okay, through translation. So this is my template mRNA molecule. So just like we saw with replication and transcription, right, that template provides the code that we need to be able to read and create the correct product. So this process, again, we're producing proteins by pulling and decoding the sequence of mRNA codons. So mRNAs have these codons. My tRNA, which is what I'm going to be using, and here's 3 examples. Here's a tRNA, and here's another tRNA, and another tRNA. Those have a sequence of letters at one end called an anticodon, and that's what you see here in step 2. So let me pause for a minute and go back up here. So step 1 again, I have my mRNA transcript. It's going to come out of the nucleus. It's going to go to a ribosome. And that mRNA, once it attaches to a ribosome in the cytoplasm, whether it's freely floating or attached to the rough ER, either way, that is kind of my step 1. So ribosome attaches and it's going to bind to that mRNA. As that happens, the ribosome is going to start to kind of scan along and move, like physically move down that mRNA molecule to find the start codon, okay? And the start codon, remember, is something that we find in mRNA here, AUG, that corresponds to the same amino acid every single time, which is here called methionine, okay? Now, how it finds that is based on pairing, and how we actually add that methionine is based on the interaction between mRNA, rRNA that makes up the ribosome, and tRNA that's going to be holding at one end an amino acid and at the other end contains that anticodon that I mentioned before. So this anticodon here, which is found, these are the 3 that are kind of sticking out as almost like a plug, right, from this tRNA molecule. They're going to bind and interact with, just through those hydrogen bonds, the codons found in my mRNA. So if my mRNA start codon is AUG, that would mean my tRNA complementary anticodon needs to be UAC, right? Because U and A pair together and C and G pair together. And they're only going to interact so long as it takes to actually take the amino acid that's up here and add it to a growing protein chain. So again, it's just temporary. And in a cell, it's going to happen, pretty quickly. It's not going to be something that lasts for very long. But that pairing is going to make sure, it's a way of ensuring that we're adding the right amino acid in the right position. If they don't pair properly here, the amino acid doesn't get added, and the cell will continue to search for the correct tRNA with the correct amino acid. Okay. So as we're kind of causing these interactions to happen between these anticodons and codons, we're also going to do some work in terms of actually creating some bonds. So I mentioned those tRNAs will stay interacting with the codons of mRNA so long as the amino acid is still attached to that original molecule. But our end goal here is going to be to make a huge protein. And so as part of that, we need to start linking together our amino acid monomers. Back in chapter 2, we talked about the special covalent bond that we find between amino acids that make up proteins, and that's called a peptide bond. So remember, covalent, it's going to be super strong. They're still sharing pairs of electrons. It's specific to proteins because proteins are that important. So one of the roles of the ribosome is not just to provide a place or the ability to kind of scan the mRNA. It's also going to use proteins that are kind of embedded in this molecule to catalyze or kind of coax that peptide bond to start forming. So it's going to start to actually create a peptide bond between those pairs of amino acids that are going to be joined together here, okay? So it's only with the help of the ribosome and the proteins and the enzymes present there that we get these peptide bonds forming. And once that happens, you can see this tRNA is leaving, okay? And then the ribosome will kind of scoot down to the next, and it'll scoot down to the next and do the same. And it continues over and over and over, and it's very organized and efficient and all of those other words that you could possibly think of because really of this anticodon and codon pairing. So it's going to continue on until the ribosome gets to a stop codon. Once it finds that stop, it's not going to add anything else. So you can see here in this picture, move myself a little bit, it's continuing on. Here's my growing polypeptide. And here are my tRNAs being pushed out as that ribosome is kind of, moving down that mRNA strand. Once it reaches this stop codon, nothing's going to be added here. There's no more tRNAs that pair with this. There's no anticodon that contains a complementary sequence. So as a result, whatever was left here gets kicked off. The final peptide bond is formed here, and then this covalent bond here is broken, and the polypeptide, aka protein, is going to be released. mRNA at that point is going to be broken down because it's just temporary. And so those nucleotides that made up the mRNA will be sent back kind of individually into the nucleus to be used to make more mRNA in the future. tRNAs that drop off and kind of lose their amino acids here in this process will remain in the cytoplasm, and they can recharge, which essentially means they're going to pick up another of the same amino acid to be used to build another protein, right? And if you remember what we said in 14.1, this is going to be kind of a continuous process throughout all of G1 and all of G2. And so it makes sense then that we want to be able to reuse these mRNA nucleotides. We want to be able to reuse these tRNA molecules as much as possible to make this process efficient enough to occur frequently in both of those stages, okay? So the polypeptide then gets released, it goes off, it folds, it may get sent out of the cell, it may remain in the cell, but how those amino acids interact with each other has a lot to do with what that protein's able to do, like what its actual function is, right? But that's kind of the whole process, right? That's what we see happening when we talk about translation. And so in my mind, I do think it's more complicated than what we saw with transcription. But that being said, it's certainly manageable, right? It's a different type of process, but keep in mind, the big picture stuff. We're talking about going from one language to another language. That's what we call it, translation. And that's why it involves so many different kind of moving parts, right? We needed mRNA to carry the instructions from the nucleus. We needed transfer RNA to carry amino acids and interact properly with that messenger RNA. And then we need ribosomal RNA and also the proteins that make up that organelle to help form those peptide bonds between our amino acids of our polypeptides, okay? So that's all I've got for 14.2. If you've got questions, let me know. Again, go back and replay that part about how you identify amino acid sequences given an mRNA sequence or a DNA sequence if you feel like that's helpful. Otherwise, keep practicing those. Watch the videos included with this because they're going to be important in helping you to kind of visualize this whole process, okay? And then let me know if you got any questions, all right? Otherwise, I will see you next time. Bye, guys.